Good morning. My name is Kevin Noe, and I'm with the Office of International Programs here at Colorado State University. Today, it is my privilege to welcome you to Session 2 of the International Symposium titled Going Viral, Infectious Disease in the 21st Century. I would like to introduce first our moderator, Greg Ebel, Professor of Microbiology, Immunology, and Pathology here at CSU. We have three outstanding panelists today, which I will now introduce. First, we have Christy Mayo, Associate Professor of Diagnostics, Vector Biology, and Virology, also at CSU. Rebecca Kading, Assistant Professor of Microbiology, Immunology, and Pathology. And Tony Schounts, Associate Professor of Microbiology, Immunology, and Pathology. I would like to remind everyone that during today's session, please utilize the Q&A box down below. Any questions that you have, please leave. And towards the end of our session, we will try and get to as many questions as possible. Thank you very much. And now, Greg, I hand it over to you.
Thanks a lot. And thanks everybody for joining today. So I'm going to start today by just saying a few words to set the stage for the discussion that I hope is going to follow. And then we'll move quickly to the panel and get started with them. So the topic is emerging diseases in the 21st century. So that's just an incredibly timely, important topic for all of us to think about, reflect on, and to discuss. Now, it doesn't take a genius to look at the world that we're living in and see that it's changing really quickly, and that we have some very big problems that currently face humanity. One of these is the emergence of new infectious diseases. Just over 20 years ago, West Nile virus was introduced into New York. It then spread throughout the US and is now this permanent fixture of our summers. Since then, we've had epidemics of SARS-1, Middle Eastern respiratory syndrome virus, Ebola viruses, and chikungunya and Zika virus, all of which have become these global chikungunya and Zika are now global mosquito-borne pathogens that cause significant suffering and death in much of the world's tropics. And of course, we're now doing everything virtual, wearing masks everywhere we go, living through this pandemic of COVID-19, the disease that's caused by a virus called SARS coronavirus 2 or SARS-CoV-2. So the details of all these different disease emergence events are slightly different, but they're emblematic of some really complex and deep problems that we as a society have to address. So there's a lot happening in the field right now. And on the panel today, we're lucky to have three quite accomplished scientists and also colleagues of mine and friends, and I hope we're all still friends after we're done today, from Colorado State, who all work on different aspects of this general problem of global emerging infectious diseases. So we're going to spend a few minutes talking with each of them about their work and how it relates to this really big, complex set of problems, and then we'll open the session to questions and discussion, and just remind everybody to put questions in the Q&A box on Zoom. Okay, so Christy Mayo, let's start with you. Can you just tell us a bit about your background and how you came to work on emerging infectious diseases?
Yeah, so thank you, Greg, for this opportunity. And yeah, it's kind of a unique place in which I started here. Basically, I'm a veterinarian by training from the University of Georgia. And I happened to wander into the micro-residency here at CSU a long time ago, 2006. And what's interesting about my journey, I think, is the curiosity and persistence of finding a case. I had a former mentor, Dr. Hannah Van Kampen, who saw that inspiration or that curiosity and really bolstered it, just offering so much mentorship. So there was a blue tongue case actually out here on the Front Range. And as a diagnostic, and that's where my role is now, the diagnostic virologist, I think it's that natural curiosity of your diagnostic cases feeding back into my research, and then my research feeding back into the diagnostics and building that program. So a blue tongue case actually got me so excited about it. Blue tongue virus being a virus that's transmitted by a little midge, a culicoides. So if you've seen gnats out on the Front Range, they're the cause of spreading this around, and it's invertebrate host. but it also affects ruminant species, so cows and sheep, and can infect them. And so one of my curiosities has been exploring that throughout my journey. I left the residency here with a master's and then went on to Davis to finish out my journey for blue tongue. And then I was lucky enough to have Dr. Van Camp, and she was retiring the year I was looking for a job, and I landed back here. So I've been really lucky to explore those paths and be able to continue some of the stuff that we're working on with blue tongue virus. And leading into what we're doing now, we've been lucky enough to be funded by the NIFA programs, the USDA, on studying the evolution and ecology of infectious disease. And so that program has given us several years of funding to really understand both the evolutionary dynamics and the ecology in this big multi-host pathogen system. And I have to say, I'm honored to be able to deliver the message today. It's a very complex system. We have a membership of about 25 people in this grant program, and it's a highly collaborative, very energized group across multiple different institutions. But we'll probably get into more of the discussion in the viruses in this panel. But that's me in a nutshell and how I kind of came to be where I am. And it's a really fun journey. So.
Yeah, so you work on blue tongue virus. Can you just tell us a little bit about the ecology of blue tongue and why it's something that you're keeping an eye on?
I sure can, yeah. So the unique part of blue tongue, it is endemic here in the US, and I guess to understand it, it's a segmented virus. And so there's a lot of fun moments to understand the evolution because it can reassort, much like what we see in influenza virus. So parts of the genome, if it's co-infecting a cell, can swap portions of its genetic material. And in understanding that with the different dynamic systems with endemic Culicoides species or gnat species here in Colorado, it gives us a platform and a model system to understand what could be emerging out there. So if anyone's heard of African horse sickness, its emergence in Thailand recently caused really dramatic losses of horses. And so with that, we try to understand this as a model system in North America for potentially understanding the emergence of maybe a different reassortant virus of blue tongue that might cause issues for our ruminant communities, or also understanding it as a model system for some other diseases and viruses that we don't definitely want to have here in North America. So really both the concepts of modeling and mitigation, trying to understand that ecology really helps us understand how we might push out better models for what we can predict.
So blue tongue virus is what's called a zoonosis, correct? So this is a virus that exists out in the wildlife. And so what you have to deal with your project on ecology and evolution is the virus evolutionary dynamics that you just told us about, but then also this idea that there are wildlife that are infected and then those intersect with livestock and then the environmental conditions. So that's like really hard to think about all that stuff. So how do you approach that as a researcher, as a scientist?
Yeah, so collaboration is the one key word, and really talking a language and learning the languages of these different communities. So when you do have wildlife and domestic species overlapping, sometimes that can be a very sticky place to be, and understanding those communities and how you get everyone compromised to the same table, understanding that we want to try to really understand the science and help all the communities in that environment. But you're exactly right. Trying to, I will say we're uniquely poised here in Colorado, Wyoming, and even Montana to have so many wildlife and domestic species interacting. And so that is part of the challenge, though, even for my field students, nevertheless, is that they're migrating long distances out into the field to collect insects and also do what we like to try to also put a layer of sentinel animal surveillance on that. So not only understanding the insect side, but also what does it look like in the animals. And so this summer we have a fun opportunity to really roll this out. Last summer was a little impacted by COVID. I have to give credit to my grad students, though. They're so eager. They got a whole field season under their belt. So, it'll be exciting. And I do appreciate you understanding those multi-layer dynamics that really come into play. But it's complex. And I think communication is also the other thing with the collaboration, is making sure that everyone's well informed of what the studies are intended to deliver.
Yeah, so that's pretty amazing that you were able to get a field season in during the pandemic. And I think also everybody who's on this panel has as part of their program field research. So do you want to just say a few words about how that's important to what you're trying to accomplish with this?
Yeah, so again, thank you for highlighting that. One of the benefits I think of all of us here on this panel are we appreciate so much the applied laboratory research that comes back to the field research, and both contributing to the common goal of just building our knowledge base. We do a lot of laboratory-based studies with a colony of Culicoides, but that can only tell you so much of what actually is occurring in nature. And so really, harnessing both capacities and designing designed field experiments, understanding that nature is also out there, always, nipping at the heels of the challenges. And so I think one of the big things that we do is having a study design that's as well developed as possible, but also recognizing that at times conditions are going to cause issues. And so that is a challenge on our daily dialogue. And so also, again, coming back to the communication and having backup plans are always really critical to where we're going with our mission.
Thanks. Tony, so let's go to you. Can you just tell us a little bit about where you come from and how you got to be here working on these really interesting emerging viruses?
Yeah, so I'm trained as an immunologist, and my graduate work was mostly in basic functions of these cells called T cells, which sort of act as controllers of the immune response. And without them, you can't really mount a good immune response to infectious agents. And my first academic appointment was at a small college in Western Colorado at Mesa State. It's now called Colorado Mesa University. And I was looking for a local research project to get some undergraduates interested in. I had just read a story about a young gal who had survived hantavirus infection. And these hantaviruses are really important pathogens globally, but they were relatively new to the New World. And I ended up deciding that we should try to focus on something that might look at the immune response of the animal that happens to be the species that hosts or in which this hantavirus resides in and circulates. And what's peculiar is, of course, that in the deer mice, which is the reservoir of this virus, the virus causes an asymptomatic infection, and there's no sign of disease at all, and they're perfectly happy with it. And unfortunately, they can't get rid of it, though. So once they're infected for life. And then for whatever reason, usually it's because they decide to build their homes and their nests in people's homes or their outbuildings. People come in contact with these rodents, and the virus gets into them, and that ends up causing a really nasty disease called hantavirus cardiopulmonary syndrome, which kills about 70% of the people it infects without treatment, and with treatment, it's still about 35% fatal. And I was kind of curious to know how is it that these rodents, these mice, can carry this virus without any disease at all, yet in humans it causes this really catastrophic disease. And so that's sort of how I decided to get involved in it. And then I had the luck of meeting a CSU faculty member at the time, Charlie Kalischer, who was trapping rodents over there in Western Colorado. And he was helpful in getting a research program started here, along with Barry Beatty. And so that's kind of how I fell into zoonotic diseases, because I wasn't really an infectious disease person. I was really just a basic immunologist. And I kind of got lucky in that I had this sort of background that prepared me to do the work. And then this opportunity presented itself. And that's how I got interested in zoonotic diseases. And now, of course, I work not just with rodents, but I also work with bats, because a number of important viruses are from bats, including this SARS-CoV outbreak. And so we're intensely studying this virus as well.
Yeah, so that's one of the things that I love the most about Tony's work, is that he's one of the only people in the world who's working with these really difficult, hard animals that that most people don't work with. So do you want to just say a few words about why that's important, and how you kind of got that sort of constellation of research interests?
Yeah, so I think it's mostly because of my interest in understanding why these reservoirs of all these viruses don't get sick, right? I mean, we hear about these nasty viruses, especially the ones from bats, Ebola, and another one called Nipah, and of course SARS and MERS coronaviruses, and SARS-2 that causes COVID. And it's pretty clear that in the animals in which these viruses circulate, they don't cause any disease. And I want to understand that, because the idea is then if you can understand what's going on in the reservoir, then maybe you can develop some new clinical strategies to help manage or mitigate disease in humans. And so that's kind of been my interest, just to really just look at the biology. How have these viruses evolved to cause this sort of innocuous infection in their reservoirs, but when they get into humans, they cause these really nasty diseases with oftentimes high mortality rates.
Yeah, so that's really interesting. So if you think about those two different types of hosts, and we're talking and thinking about this problem of infectious agents that at one time in the recent past were confined to these little tiny geographic areas, and then they explode. And you can think about SARS, you can think about West Nile, and they entered these new types of ecosystems. It's a huge problem. And so I was just rereading your really interesting paper on SARS-CoV-2 infection in deer mice, and the conversation that started about this idea of reverse zoonosis, which is was something that I hadn't really thought about. Do you want to kind of talk about
Sure.
what that is and give us a few take-home messages from that paper? It was really an interesting one.
So here's the main issue is that this virus, SARS-CoV-2, the one that causes COVID-19, this pandemic that's ongoing right now, this virus and many viruses that are very similar to it circulate in a really large number of bats in Asia. And it's not just China, it's many places in Asia where these viruses are found. And so that virus somehow managed to get from the bat into humans in China. And we're not sure of how that route occurred. But nonetheless, it got into humans, and it has been adapting quite nicely in humans, apparently, because of all the new genotypes that we're starting to see. So here's a key feature, is that this virus is able to transmit person to person really efficiently. Most of these zoonotic viruses that we work on don't transmit person to person. Either they have to evolve a way of efficient transmission via person to person routes like respiratory, or in the case of the mosquito-borne viruses or other arthropod-borne viruses, you have to have a vector that's capable of transmitting them as well. And that's what we saw with West Nile virus. So then the concern is that what happens if this SARS coronavirus 2 manages to make its way into wildlife in North America? Can it then establish itself as a permanent residence here, just like West Nile has established itself as a permanent residence in North America? And so we did a study where we inoculated some deer mice that we have in a colony here at CSU with this virus. And sure enough, not only do they get infected, but they're able to transmit the virus to other deer mice. And that is a big factor for sustaining the virus in North America permanently. Now, there are a lot of other things that have to happen. For example, the virus has to sort of establish a way of spreading either fast enough to maintain itself or infecting the deer mice for a longer period of time. And those haven't happened yet. We still don't know if it's gotten in the wild yet, but I tell you what, with all these mink farms in many states in the Western United States, especially in Utah, where there have been more than 10,000 minks that have died after contracting SARS-CoV-2 from their human handlers, it's pretty clear that in those mink farms, the barns that house those mink are accessible to deer mice and other wild rodents that are found in North America. And it doesn't take too much of a stretch, in my view, that if those rodents get into these buildings and these mink are kept in these open wire cages where the food can drop, the feces can drop, and we know SARS-2 can be transmitted that way, or at least shed that way, then that's not too far of a stretch in my mind for the rodents to become exposed to the virus. And if that were to happen, then there's a chance that this could then become a permanent resident in wildlife in North America, particularly the rodents. Then it doesn't become a stretch to me that, if you live in a rural community and you have cats, and we now know that cats are susceptible, and one thing cats like to do is catch rodents and bring them in and show them to their owners, and they drop them on their front porches. And now you have a potential transmission system between a rodent and a human via their cat. And this isn't without precedent. We've already talked about West Nile virus, but there's another coronavirus called Middle East Respiratory Syndrome coronavirus, which also originated in bats, but it's now established itself in camels in North Africa and the Middle East. And now almost all human cases are because of transmission from camels that then lead to these outbreaks that we see every year.
Yeah, so after Zika was first introduced into the Americas, there was several groups of scientists, researchers who were looking for that sort of spill back from a human mosquito cycle into a cycle with non-human primates and mosquitoes in the tropics. Are you aware of anyone actively looking for SARS-CoV-2 infection in wildlife in America?
I know of, so I'm not sure what I can talk about, but I know that there have been some groups looking at rodents in Utah and also in mink, because apparently these mink often escape from their barns and get into the wild, where they can then encounter other wild mink. And it seems that there's been at least one report of a wild mink getting this virus. And so the question is, how many other mink are getting infected and getting from these captive mink, and is that going to lead to a permanent residence of this virus here? And I don't know the answer to that. We've tried to get a permit to collect rodents over in Utah, and we haven't had any success from the state of Utah to get that permit yet. It's not that they've said no, they just haven't responded, and that makes it a little more difficult. And I'm sure it's because they're swamped with other things and pressed for time because of COVID restrictions as well.
Yeah, once these things become established in wildlife, it sort of changes the whole calculus of how one has to think about them.
Yeah, but I think a lot of different rodent species in the Americas are susceptible to this virus. I bet it's in the dozens at least.
Okay, great. Thank you very much for that. So Rebecca Kading, let's get your story. How did you come to be working in the field?
Oh, well, I guess I'm one of those stories where I didn't think I would end up here. It was a very circuitous path, but I'm very thankful to be working on the viruses and disease systems that I'm working on now. And I think in hindsight, everything has kind of come together in a really cool way. I guess going back to college, I was very interested in wildlife conservation, and attending the University of Delaware at the time, they didn't have wildlife conservation as its own major. They do now, but at the time it was kind of couched within the entomology major. So I was forced into the bug classes in order to study birds and mammals. And I sat in the back with my arms crossed, and I didn't want to be there. And then it turned out to be really interesting. And so then I ended up, the summer after freshman year, and this would have been pre-West Nile, but they were still, I got a job with the state of Delaware mosquito control program. And Delaware being a coastal state, a lot of their nuisance mosquito and disease surveillance, or they're just still doing surveillance for Eastern equine encephalitis virus, was focused on salt marsh mosquitoes. And so there I was in the helicopter and in the boat doing surveillance out on the National Wildlife Refuges and getting to birdwatch to my heart's content. And then on my lunch break, I started reading some books on yellow fever and all these viruses that mosquitoes carry, and that really piqued my interest and kind of got the wheels turning in my head. Like, all these systems are interconnected. There's mosquitoes out there transmitting these viruses, and there's birds involved, and there's this is really fascinating. And so then that really kind of set my trajectory to learn more about the role of insects and pathogen transmission, but also how things are all interconnected, and that, we have to have kind of a holistic view of this and understand the reservoir side, the pathogen side, the environmental side, the insect vector side. And so that's kind of, shaped the way that I've taken my career.
Yeah, that's great. So in your lab now, you're working on something called Rift Valley fever virus. And to me, this is like one of these viruses that everybody's terrified of, but you're probably one of 2 or 3 people in the world who are actually working on it. So could you tell us a bit about the virus and why it's something that that you're paying attention to, and why we should all be paying attention to it?
Sure. So Rift Valley fever virus, it's a bunyavirus, and so it, just like the virus that Dr. Mayo is studying, this is a segmented virus, so it has multiple genome segments. It was first recognized in East Africa in the 1930s, and it's not in the United States, but it's one of those viruses that has very high consequence to human and animal populations, and so we're keeping an eye on it as far as something that we hope is not introduced here. It has so it can be transmitted by mosquitoes, but people can also contract it directly by handling infected animal tissues. It has very high mortality rate in livestock. So it's a hemorrhagic fever virus that's pathogenic to people and animals. So if it is introduced, it would be it would have public health, veterinary health, food security, economic. It would kind of hit all of the sectors of our economy and government. So so my lab is very interested in learning some of the key, trying to find some answers to some of the questions that would be absolutely necessary to jump on immediately if this virus was detected in the U.S. So, do the mosquitoes in the United States, can they transmit Rift Valley fever virus? We're looking specifically at the vertical transmission potential in mosquitoes. So can this virus be transmitted from a female mosquito to her progeny? Because that would be another way that that could establish itself in nature. And which mosquito species would be important for this, because that would lead to surveillance and mitigation strategies if it's detected.
Yeah, and so are you in your lab now? Are you doing? Do you have some field work that you're involved with around this kind of effort?
So right now we're kind of taking what we know and what we don't know as far as we as the scientific community about the ecology of Rift Valley fever, and trying to answer some of those questions in the lab. And so one of the hallmarks of Rift is that it has these cyclical outbreaks that seem to follow climate patterns, and so particularly with El Nino events. And so you get a big outbreak of Rift Valley fever and every 10 or 15 years, and then you don't hear from it for a while. So what's going on in that interepidemic period? Well, it's there. It's still circulating at pretty low levels. I think that the vertical transmission by mosquitoes, the mosquitoes themselves are serving as a reservoir for the virus to some extent. That explains some of it. So those are the types of questions that we're right now trying to answer in the laboratory. We would like to get into some field studies in Africa to see what's going on and during the interepidemic period. In the meantime, our fieldwork efforts have been focused here in northern Colorado just to look at the, kind of under a hypothetical scenario with the agricultural, the livestock production practices that we have here and the mosquito vectors that we have here. And if you were to introduce this virus into the system that we have here, what would that look like? And what would the risk factors be? What mosquito vectors would be involved? So try to think about it in those terms, in preparation for a potential introduction.
You also have some field studies in Africa, right?
I do, yes, and that also those are focused on bats and looking at human-bat interactions in Uganda.
Yeah. So one of the other things that I really like about what you're doing is your work around on transboundary infectious diseases. I mean, I think this is something that all of us are thinking about a lot more right now. And I mean, you can think about it with Rift or kind of any of a fairly wide array of viral pathogens. So, I mean, what's that work been telling you about what we need to do in that area of transboundary health and passing viruses around across boundaries?
Yeah, it's an excellent point. I think the lesson that we learned is from West Nile, Zika, chikungunya, Ebola, SARS-CoV-2 now is that these viruses move, that there's no, and they can move quickly with movement patterns. And so it's important to think about this on a global scale and how quickly one thing can move from one place to the next. And so, kind of looking at the risk of introduction and what things would look like if they're introduced into very different ecological systems and how to combat that. And I think that, there's a lot of layers of complexity as far as how this is dealt with and mitigated and strategized, and because it's very it's very complicated. But I think that it's not looking at something emerges here and moves here. It's like we're all at risk for everything, so we just need to be prepared.
Yeah, that's great points. Tony, I forgot to ask you about whether you have ongoing field work. I think that you do. You want to talk about that?
At the moment, we don't. We have, we're ready to go. We can't do anything without permits, and so consequently, we're sort of on hold right now. Plus, with the weather, rodents are pretty dormant for most of the time during the winter. So we probably won't get out until April. We do some field work in the Caribbean with some bats. We're looking at influenza viruses there, but again, no travel right now because of COVID. So hopefully next year this time we'll be down in Grenada collecting bats again. So
great. Okay, so now let's just take a step back. The three of you on the panel are all working really hard in the field, and we're making new discoveries, pushing science and sort of this field of emerging infectious diseases research forward. And I think that what's clear to me just listening to you talk is that everybody's got a slightly different perspective on your work and on this kind of general problem. So if we go back to where we started, to this idea that we're living in this world that is beset by emerging viruses, and of course, SARS-2 is the one that we're most obsessed with at this particular moment. I just want to put the question to you before we open the questions more generally is, what the heck is going on, and why have we seemingly gone in this really short straight line from West Nile virus to Ebola to SARS to CHIK to Zika to SARS-2? What is kind of happening in the world that's leading to that? And then we can follow up on that all day long if we want to, but I'd like for all of you to just kind of weigh in on what you think is going on and where this all is heading.
well, I'll jump in. So I think there are a lot of things that are sort of conspiring to lead to all these changes. Of course, emerging infectious diseases are nothing new. They've probably been happening, for as long as humanity's been around. But I think what's different is that there are a couple of really important factors that are contributing to it. First of all, the world is becoming much more mobile than it used to be. And so I'm certain 20 or 30 or 40 years ago, there were outbreaks of infectious diseases that we didn't even learn about because they were local and they were contained and they went away. But now it's very easy for somebody to get on an airplane from, for example, Asia and end up in the United States within, 18 or 20 hours. And if they're carrying something with them, especially something that can be transmitted person to person, then that's going to lead to a potential outbreak in remote locations like we've seen with SARS-CoV-2. I think the other big issue is that climate change is really having an impact, and it's going to accelerate over the next several years. And I think those factors all contribute to the emergence of these infectious diseases because climate change is a really important driver of how these viruses travel. And then finally, of course, humans are always encroaching upon wildlife habitat. We're building new buildings and cities and civilization. And when we do that, we inadvertently run into animals that might be harboring some of these infectious agents, which can then get into people either directly or via animals that serve as intermediate hosts. And so to me, those are the three big things that I think about. I'm sure there are others that I'm missing, but to me, those are the conspicuous ones.
Christy, Rebecca, what do you all think about that?
I agree completely with all of those. I was thinking that the human animal interface is shifting at some point, but just in thinking more broadly about the concept of spillover and movement of an infectious agent from a natural system into something that's SARS-CoV-2 and transmitting very easily from person to person. There's a lot of things that need to happen in order for it. I mean, that's just such a, there's such a big stochastic process, and there's a lot of barriers to that happening that, in this case, those barriers were overcome. But in just, going from an infectious agent circulating in an animal population and all of the dynamics involved with that. And then how does it get into the environment or get into, to get there's that interface between the environment and the animal and the pathogen. And then how does it get into people? And then there's barriers within us too, with our susceptibility to it. And then it's kind of a big deal for something to go from person to person rather than have it, come from an animal. So there's so much going on that, if one of those things gets tweaked and for whatever reason, then that makes it easier for spillover to happen. I think that the reasons that Tony gave are kind of increasing the susceptibility for that to happen by increasing our contact with these agents in some way.
And I would echo that very much, both of your comments. And I spend a lot of my time thinking about climate variability or thinking about the next grant we're going to write, correct? And some of the things would be definitely the climate variability for expansion of some of these, especially with the vector-borne space, and then also just within what is endemic now, what the dynamics and shifting patterns, echoing everything that Rebecca and Tony have said. The other thing that I, just as a diagnostician and veterinarian, is the advancement in our technology. Right now, when something may not hit a target in terms of what somebody has designed, maybe a kit or a PCR, we can now adapt or adopt next-gen sequencing or next-generation sequencing and be able to explore the unknown, knowing that we tried to target every pathogen we thought might be on that list. But this, I think that contributes to the diagnostic space I live in, but also my research interests. When maybe viruses don't quite fit in the pigeonholes I want them to, we find new opportunities. And so next-gen sequencing to me, and probably more technology that's going to unveil and probably has already unveiled, I think that's giving us a lot of latitude for Tony's comment that potentially there have been outbreaks or emerging diseases that we just maybe haven't seen before or haven't noticed or taken note of. So, and then I can't help but also plug for the epidemiology portion of this is just enhanced surveillance programs. I'm designing the new technology to harness, programs that are meant to actually understand what's currently going on out in nature. So yeah, I think that's kind of the enthusiasm I have moving forward on some of these issues.
Okay, so we're starting to have some questions come in from the audience. I'll just remind everybody, please put your questions in the Q&A and they will get relayed to me and then I'll relay them to the panel. So here's a question for Tony. Dr. Shounds, do we have any clues on anything that bats or mice have that we can replicate in humans to make their diseases innocuous as well? For example, temperatures in bats seem to play a part in keeping their diseases at bay.
So I'll address that last part first. I think this temperature, so the idea is that because bats fly, they have elevated body temperatures from their metabolism. That somehow mimics what some people call the fever response that controls infection. And I think that's totally bogus because by the time you get a fever during an immune response, there's already a lot of other things going on. An elevated body temperature from a fever response is quite different than just generating body heat because you're exercising or burning a lot of calories because you're flying. So physiologically, they're two completely different things, and I'm not a fan of it. And I just want to point out that there's only been one study that's been done to look at the effects of temperature on, in this case, Ebola virus replication. And it turns out that if you elevate the temperature of cells that are infected with Ebola virus, it doesn't replicate any better or any worse. It's just the same. And so I know that's just a sort of a laboratory study that doesn't look at an animal, but it's the only thing that's out there, and that would argue against this sort of idea that elevated body temperature has some impact on the bat's ability to control these infections. What I would point out, though, is that it sure seems like that bats have a really, really good innate immune system and this is the system that is sort of always in place, always looking out for infectious threats and dealing with those threats. And their adaptive immune system doesn't seem to be as important. I'm not saying it's not important. I'm just saying it's not as important as the adaptive system is for humans and mice, which is what we teach about in immunology. You know, in all of our immunology courses, we talk about two mammals really, the laboratory mouse and humans. And there are 6,000 species of mammals out there, and they're all going to be a little bit different. So I think that may be an important difference for the bats, why they're able to have these viruses. But I want to point out that the viruses have to adapt to the bat system or whatever reservoir they're in. Because if they don't, they either get cleared, sterilizing immunity, or they cause disease in that animal. And that's not how a reservoir host relationship works between an animal and a virus. This could very well shape why these viruses are so nasty in humans, right? Because if you're a virus and you've evolved in this system that has this really robust innate immune response, and then you get into a human where the innate immune response is okay, then suddenly that virus may have a competitive advantage over those cells because the cells are ill-equipped to deal with the virus that's evolved in a high innate immune response environment. So that's kind of the angle that we're working on right now. But again, it becomes really difficult to experimentally address these questions simply because we don't have the tools and technologies to manipulate bats and like we do laboratory mice, right? We can go in to a laboratory mouse if we think a certain gene of the immune response is important for some viral disease, and we can go in and easily disable that gene in a mouse and look at the outcome. We can't do that in bats yet, but I'm hoping we get there within the next couple of years or so.
Okay, there's one here for Rebecca Kading. The questioner appreciates what you said about connections in your work and is curious, does this relate to biodiversity? How does your knowledge of biodiversity influence your work?
That's a great question. Yes, I think biodiversity plays a big role in all of this. Certainly that, in some of our experiences in Uganda, when we're looking at, and also in Central America, I've worked in Guatemala too, very ecologically rich locations, very, very biodiverse locations. It seems like, when we start seeing this, contact with human infections, that there's some reduction in biodiversity that relates to the increased contact with either a direct contact pathogen or a vector-borne pathogen, and certainly with the mosquito-borne viruses and surveillance that we've done in Uganda, we're picking up more viruses and vectors at that kind of that interface, that ecotone between very diverse systems and very simplified urban settings. That's kind of where that was happening. So I think that and another example where biodiversity has come into play with, we have a mosquito species called Aedes albopictus, Asian tiger mosquito, and Aedes aegypti. And Aedes aegypti, feeds almost preferentially on people. Aedes albopictus has a much more diverse blood feeding behavior, much more opportunistic. And so, but both of them are very capable of transmitting viruses, the same viruses in the laboratory. But you have to consider the ecological context. And Aedes aegypti, will be driving transmission of Zika and chikungunya, yellow fever, kind of in urban settings. Aedes albopictus is very capable of transmitting those viruses, but if it feeds on a frog or a dog or a bird, then that kind of dilutes its effect in the natural system for contributing to transmission in outbreak situations. So yeah, biodiversity is very intertwined with all of this.
So your response to that question just leads me to another question that I'm going to skip forward to. We'll get back to as many of these as we possibly can. The question is, with Aedes and Anopheles mosquitoes creeping northward deeper into the US, what should governments and society be doing now to prepare for dengue, chick, Zika, and malaria? So that's part one of the question. Another question that was asked earlier that I want to tie to this one is, how is climate change, because you all mentioned climate change, you all mentioned climate, how does that really affect this sort of acceleration in emerging infectious diseases? So start with the Aedes and Anopheles question and then the climate one. So maybe Rebecca, do you want to take the question about what we do to prepare for these things? And then Christy, can you talk about temperature and climate? Okay.
To prepare. So certainly, surveillance would be a big component of it, and looking at the range expansion of the vector. and then any geographical differences there might be in the susceptibility of the vector transmitting, what would happen. I think there is some, upward limits to Aedes aegypti as far as overwintering, that would affect things. But I think, as far as, preparation there's this vector control component for these vector-borne pathogens, and there's therapeutics and vaccines, and we just kind of keeping an integrated approach towards everything will be critical.
And then to speak to some of the climate, I like to use the term, and I'd love to just have everyone weigh in on this with me, but I like the term climate variability because I think with the changes of, in consultations with some of the climate folks, it's, I guess the Al Gore model of just steadily warming, there's probably going to be some turbulence along the way as we do warm in the globe. And I think the context of climate, yes, I think we're going to have expansion, and I think there's going to be differences among what dynamics. Rebecca mentioned El Nino, La Nina, all these different dynamics that are really contributing globally to the changing distribution and predictions. And so in my space of modeling, I like to hang out and think about what parameters need to go into this model to make the best prediction, but that's often hard to really conceptualize when there is so much variability. And the context to that, just coming back full circle, is the influence not only on the vector populations as adults, but also their larval habitats or their habitats in general, and what these different dynamics of rain or humidity or temperature. It's a really complex system that's not only based on just, again, that temperature alone that we monitor. So, I know both of you think about this in your spaces as well, but just to also add what your thoughts are on that.
Well, I think there's, in terms of the, just thinking of temperature, and my brain goes to some of the, the studies, and many of them done in Greg's lab too, just looking at, kind of the role of temperature in virus infection. And, but there's also effects on vector survivorship on the upper limits of that as well. And so, so I think that it's going to be extraordinarily complex. I mean, there's definitely a linkage between temperature and the dynamics of the virus infection, and how quickly the mosquito would be able to transmit after that. But then there's some limits to that as well, as far as will the mosquito then survive long enough to transmit, or is it there's optimal ranges. And so yeah, I think there's going to be a lot that goes on.
Yeah, there's a lot of layers there you have to kind of work through, because temperature affects pretty much everything, and not just mean temperature, but temperature variability. There's all kinds of ways of thinking about that. It's really, really hard to pin down. Tony, there's one here that I think you probably are in the best position of all of us to answer. Are we seeing advancement in antiviral drugs to expand on our currently common Valtrex and Tamiflu? Can more broad spectrum antivirals be developed?
Yeah, so this is sort of the holy grail of virology, and
Yeah.
I don't know of any that are even close to being available. And by broad spectrum, I'm using the term like we use it for bacteria, right? So against both gram-positive and gram-negative bacteria. And viruses they're much more diverse in a lot of their biochemistry, and so that seems to be the Achilles heel of developing a broad spectrum antiviral. So I'm not optimistic that'll happen. There might be some broad spectrum anti-influenza drugs that are coming out. I mean, there's one now that's really amazing because it interferes with a common biochemical feature of all influenza viruses, and it is able to knock down all those viruses that have been tested. But once you start moving outside of a family of viruses, then it becomes much more difficult to develop broad spectrum, so to speak, broad spectrum antivirals. So I'm not optimistic about it, but again, it's not my area, so there could be things out there that I don't know about.
Yeah, I agree. I mean, typically antivirals target these really specific biochemical pathways, and that's why they're safe. Yeah. So it's a huge challenge to develop something that would be seen as suitably broad spectrum. That's my opinion. Okay, there's a There's a few other, okay, so this one was directed to Rebecca, but I think all three of you probably have a perspective on it. What implications has COVID had for research or interest in neglected tropical diseases? Has this awakened the world to care more or bring more awareness to the need for additional research, additional funding? I think it's a really interesting question, right? Because big picture, I mean, what's sort of has happened is everybody's focused on that. Is that, you know, is that a diversion, or is it a diversion that's actually positive because it sort of draws attention to, again, this kind of bigger context of the problems that we're facing as a society? So Rebecca, we can start there, but everybody please weigh in.
Yeah, I had two simultaneous thoughts, and the first one was, well, yes, it's drawing attention to, and I mean, it's hard to know what's going to be where you should put your attention. So, you know, we are diverting resources into a very important, in a very important way, positively, but also that, you know, that everything else still hasn't stopped happening while this is going on. Like Christy mentioned, the African horse sickness introduction to Thailand, and there's been, you know, there's been all these things happening that are continuing to happening that, you know, I hope that we're not caught off guard in another way while our attention is focused on SARS-CoV-2, because it's not the only thing that's going on in an NTD world. So, so those are my two thoughts.
Yeah, no, I'll just echo, I guess. Sorry, Tony. But it is going to be bringing attention that would be, you know, harnessing a pretty powerful influence of how fast this, you know, just went through populations. And thanks for bringing African horse sickness back to the picture. But, you know, recognizing we have laboratory data to support that there are competent vectors for virus combinations all, you know, in our laboratories. And the unknown is, what does that look like when you're out in nature, which I guess bolsters the field work and lab work component, really partnering very well with each other. And I think in all of our laboratories. And but I'll turn it over to Tony. But yes, I agree with everything Rebecca said.
So I stepped out a minute because I wanted to get this book off my shelf. You know, we used to fund expeditions, what I call viral expeditions, to go look for new viruses. And I don't know if that's, you guys can see that. Yeah. This is the arbovirus catalog, and every page is a different virus. And the government used to fund scientists to go find out what's out there. And they don't do that anymore. It's considered a fishing expedition. And unfortunately, there are a lot more viruses out there than just these. And these are just the ones that are transmitted by arthropods, right? You know, insects, ticks, things like that. Just to give you an example, SARS coronavirus, we've had 1 and 2 so far in the last 20 years. There's well over 100 SARS-like coronaviruses in bats that we know about. There are probably hundreds more that we don't know about. And so what do we do as a society? Well, we wait until there's this big pandemic and then we react to it. So my view is we should be more proactive, and maybe that would reduce our need to be reactive. But I don't control the purse strings, and people at the funding agencies don't seem to be interested in this anymore. Maybe this will change their opinions.
Many of those viruses are right here in Colorado, and that's right here in Colorado. This is not a.
I know. So
it's solely an international problem.
Right.
Okay, so just related to that point, Tony, I'm just going to bring this one up. Do you think policies to combat human encroachment on historically rural areas could help reduce the spread of vector-borne diseases?
Yeah, but good luck getting that done. I mean, we have competing interests, right? There are those of us who know what the right decisions are, and then there are those of us that don't know and don't care, and they have the money, and so they do whatever they want. So,
yeah,
I know I'm cynical sounding, but that's just kind of my perspective on it, right? We've seen it with this SARS outbreak. We know what to do: wear a mask, keep your distance. We know that works, right? I mean, look at the number of flu cases this year. It's going to be at an all-time low. Normally by now there are 25,000 dead Americans from influenza. And I think this year there's maybe around 100 or so. And it's not because flu suddenly disappeared. It's because people are wearing masks and they're distancing and they're not traveling as much. And so I just can only imagine what would have happened with SARS-CoV-2, COVID, if we hadn't implemented those strategies, I think we would have been far more than 500,000 dead Americans by now. So, but that's just my view on it.
Yeah. So there's an interesting question here that I'm gonna bring to the group. All of us work on dangerous pathogens. So there's a request that we discuss the risks of doing this type of research, perceived and real, of the virus escaping the lab. And I think it's important that we talk about this where we don't have too much time left. It came through pretty recently. There's been a huge amount of press, as you know, and conspiracy theories about the SARS-CoV-2 as a release, either intentional or accidental, from a lab in Wuhan. There's no evidence whatsoever to support that. I think it's important for us to just address it here because it's something I know that we spend a lot of time thinking about and a lot of time working to minimize that risk. So who would like to start with that one? I mean, I can say my piece on this for a long time, but
yeah, what we do is inherently risky, but without it, there'd be a hell of a lot more dead people than there are now. So, I mean, we take a lot of measures to mitigate an event that might lead to the virus getting out. We've been pretty good at it. I mean, if you look at the history of infectious disease work, at least in the United States, we've done a pretty good job, not so much in a couple of other countries that I won't name, but the trade-off is if we don't do this work, we're going to get, as Rebecca said, caught off guard and something disastrous is going to occur. And we're kind of victims of our own success, right? Because there haven't been these really bad, nasty things other than now we've got COVID. And last century, we had the Spanish influenza before we even knew what virology really was. And that's because we're doing such a good job at getting an idea of what's out there and how we can combat that. I'd much rather know how to deal with a familiar foe than an unfamiliar foe. And that's why we do this work, so we can familiarize ourselves with these foes that are out there. That's just my perspective.
mm-hmm.
Yeah, definitely. The consequences are too high to not study these things and to do it, not in a pandemic situation where there's urgency and flustering, and I mean to take our time and to understand these things. And biosafety and biosecurity are at the forefront of all of our minds in conducting these studies. We have an excellent biosafety team at CSU that helps think through all these things, thinking about personal barriers and facility barriers and all of these containment steps that we all take every time we step foot into the lab to make sure that we're doing these things safely as possible.
I think a lot of times there's a misperception out there that as scientists, we just dream up a study and we go and we then do it. All of our experiments are subject to a tremendous amount of vetting and a tremendous amount of oversight, not just at the university level, at the state level, at the federal level. Mm-hmm. We're subject to audits and inspections that can happen pretty much any time. CSU does a fantastic job of biosafety and biosecurity.
Yeah.
Okay. I think that's probably the last question. Kevin, do you want to return here?
Yes, so thank you very much to all of our panelists today, Greg, Rebecca, Christy, Tony, a very enlightening presentation. Thank you so much to our audience members and everyone for your questions and participating in this event. After this session ends, there will be a survey sent to attendees. If you have a moment, we would greatly appreciate if you fill that out. And if you'd like to view this session again, these sessions have been recorded and will be available both on our International Programs website and on YouTube. So thank you again to our panelists and to attendees.